Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we will discuss our last topic of pharmaceutical biotechnology, which is immobilized enzyme. So today is also our last lecture of the 10th semester. तो आज का हमारा टॉपिक जो है वो फ्रॉम द फार्मास्यूटिकल बायोटेक्नोलॉजी लास्ट लेक्चर है। तो उसमें हमने की टॉपिक्स ही पढ़े, यानी कि बेहतरीन दो तीन टेक्नोलॉजीज ही पढ़नी थी हमने, जो कि हमने डिस्कस की हैं। तो वो उन 2 से 3 टेक्नोलॉजीज में एक टेक्नोलॉजी थी जिसको हमने कहा कि वो रिकम्बिनेंट डीएनए टेक्नोलॉजी है, वो हमने डिस्कस की है। सेकंड हमने फिर टेक्नोलॉजी डिस्कस की है जिसको हमने कहा कि वो हाइब्रिडोमा टेक्नोलॉजी है। इस तरह अब हम थर्ड एक टेक्नोलॉजी डिस्कस करने लगे हैं जो कि हमारी इमोबिलाइज्ड टेक्नोलॉजी है, इमोबिलाइजेशन टेक्नोलॉजी है। तो इमोबिलाइजेशन टेक्नोलॉजी में आज का हमारा लेक्चर जो होगा वो इमोबिलाइज्ड एंजाइम से होगा। तो इमोबिलाइज्ड टर्म जो है वो मोबिलाइज का एंटी टर्म है। तो इम से मुराद नॉट एंड मोबिलाइज्ड मींस मूविंग। तो नॉट मूविंग और नॉट गेटिंग away from the reactants and the products, become remaining localized in the solution where we have added the enzymes. And enzymes are actually the proteinous molecules, protein molecules which are used to normally accelerate the chemical reactions of the living cells. Normally it is said that if there are no enzymes, there will be no chemical reaction in the body, and the body cannot produce any kind of the precursors from the enzymes or any kind of metabolic activities and anabolic activities it can produce without enzymes. So enzymes are actually those protein molecules which are used to speed up our biochemical reactions, and without those all chemical life processes would be too slow even to carry out. And enzymes, these are the specific proteins which show great specificity to the specific regions, to the specific reactants, and they are not permanently modified. They are recoverable as such as we have added to a reaction. So due to these two properties, one is its specificity and the second one is recovery, that they can be recovered by body, by biological process. They can be recovered without any modification, without any permanent modification. Means that there should be a slight modification which is reversible. A reversible modification can occur, and that modification will be reversed. Since there is no chemical, no change during the chemical reaction, so the enzymes can be reused. Enzyme can be used. One enzyme can be used for multiple type of reactions. So it is a cost effective to use some enzymes. So enzymes, when they are in the solution of the reactants, it converts the reactants to the products. But enzymes from those solutions are difficult to extract out in vitro conditions. In vitro conditions in which we are using some enzymes and then we are converting the reactants into products, and then we are retaining back those enzymes. So it is very difficult to recover those enzymes from the solution phase. The solution is that we can attach the enzyme to the reactors or to a fixed place at which it can be used again and again. We can add it in a solution or we can remove from the solution. If we add the solution, it can convert the reactants into products, and when the products are reached, we can remove that reactor from the solution phase. In this case, we can protect the enzyme and we can reuse that enzyme. This term of protecting an enzyme or making it stationary or unable to move will be termed as immobilized. So we can define immobilized enzyme as the enzyme which can be physically attached to a solid spot over which a substrate can pass through or can attach to it and convert to a products. So a physical attachment of an enzyme to a solid spot, and that can be added to a substrate and convert the reactants into products. So there are multiple benefits of the immobilized enzymes. There are first of all, we can multiply or repetitively use a enzyme system. We can pass the reactants over the enzyme, and we can remove the enzyme from the reactants, and we can use another reactants to convert to products with the same enzyme. Enzymes become stabilized due to the bonding or entrapment. Also, we have two methods of stabilizing an enzymes. We can bound to a sport by using covalent bond or ionic bond, or we can entrap it within the polymeric system in a polymeric or a gel like material. So if we can entrap it or bound it to a sport, we can reuse it repeatedly, and so the products will not be contaminated with the enzyme, and it is especially useful in the food and pharmaceutical industries. Method of immobilization will result in the enzyme to be attached to a sport or to be bound inside a polymeric gel or to be attached with the crosslinkers. So first of all, the major problem with the enzyme, because they are highly specific and the enzyme activity depends on their binding site. If we have a knowledge of the binding site or the active site at which reactant, for example, this is a binding site and here are the reactants. If the reactants are attaching to the binding site, so the activity of this binding site is necessary with the to be functional when it is immobilized. So if we have a knowledge of this binding site, better knowledge, we can protect this binding site during binding. For example, we cannot attach the sport over here. If we attach the sport over here, so there will be no place to attach the substrate in it. It will be blocked. So binder, the reactor should be attached to somewhere else other than this active site, so that the active site can be protected and active site remain functional, that it can convert the reactants into products. So one of the method in which we can protect this active site is the spacing group. Spacing group that can be attached to this enzyme's backside to make a distance between the attacher or the sport and the enzyme. So the spacing groups can be used of the carbon-carbon chain, for example, for methyl groups. So if we can attach 6 CH2 groups, that can place a sufficient type of spacing that will help to protect its active site. So the spacing groups are the groups that separate the enzyme from the backbone of the polymeric spore. Usually the spacer groups are 6 carbon groups or more than 6 methyl groups. And the second technique is that we require to avoid reaction which is on the site of the binding site. We should avoid the reaction at above, at below, or at the forward side of the binding site. So alternatively, we can protect this binding site by attaching some groups with those who are known as protective groups. Normally the inhibitors of the enzymes are the groups that can specifically bind to an enzyme. So if we can add a substrate which is a competitive inhibitor of the enzyme, it will attach to the enzyme and it will actually inhibit this enzyme or active site, and then we will proceed the attachment process of this enzyme. After the attachment, when it will be when this enzyme will be added to the reactants, it will compete with this substrate and the substrate available. So if that substrate is more suitable for the enzyme, this will compete with that substrate and substrate will be converted to the products. So there are commonly used 3 strategies for immobilization of enzyme. One is carrier binding, second is cross-linking, and third one is entrapping, as we have discussed earlier. In the carrier binding, we will bind the enzyme to a water-insoluble carrier or water-insoluble sport. It will be enzyme sport, and enzyme sport will be bind to the enzyme. Sport will be binded to the enzyme through a spacing group. A spacing group will help protect its active site. Second method is cross-linking. We can cross-link the one enzyme with another enzyme. So a crosslinker can attach to the back of the enzyme or to the back of one enzyme and to the back of the second enzyme. So enzyme can be cross-linked bifunctionally or multifunctional agents. It can be attached to another enzyme, can be attached to one or two or three enzymes to a cross-linker. So it is multifunctional cross-linking. In the entrapping, we use some semi-permeable gels, or we use some a polymeric membrane which is semi-permeable. Semi-permeable, we actually know that semi-permeable is the permeability in which a membrane is permeable or gel is permeable to some selective agents, not to all the components of the solution. It will be only to be permeable to a selective portion of the solution. So a semi-permeable polymeric membrane will be permeable to the reactants, will be permeable to the products, but it will not be permeable for the enzymes. Enzymes can be entrapped within the semi-permeable polymeric membrane, or it can be entrapped within the semi-permeable gel lattice. So it can help in removing and adding repetitively to the solution, in which we can convert it from the to the products. So first of the method in the three methods which are used to immobilize enzyme is carrier binding. Carrier binding is the most widely used and the most oldest technique of binding the enzymes. In this case, we have to attach the enzyme to a carrier or to a sport, and this attachment of the enzyme depends upon the nature of the carrier. So carrier, if the carrier is suitable for enzymes are actually proteins, and proteins can bind to polysaccharides. So a polysaccharide type of a polymer can be selected as a carrier. Otherwise, if there is something other than the polysaccharides or carbohydrates, the proteins cannot attach to that substrate or that polymeric material. So particle size of the carrier depend heavily depend on the attachment process. If the particle size of the carrier is optimum, it will stably attach the enzymes. If it is variable, for example, very small particle size and very large particle size, it will result in the random type of attachment. The surface area available of the carrier will result in the easy binding of the enzyme to any place of the carrier. Secondly, enzymes are the proteins. So protein binding to a carrier will depend on hydrophilic and lipophilic balance properties. So the molar ratio of hydrophilic to lipophilic groups which are attached to the carrier will result in the binding of the protein. For example, only the polar groups actually react to the proteins. So proteins will be reacted to the hydrophilic group, and the back chain, which is insoluble in the solution and provide a support, are the hydrophobic groups. So hydrophobic to hydrophobic group ratio is the most important for selecting a carrier binding. And then the chemical composition is most important. If there is an increase in the ratio of hydrophilic groups, and the concentration of the bond enzyme will be increased, or the activity of the immobilized enzyme will be higher. So we will have to select a specific carrier which is having some groups which have excess groups of hydrophilic binding sites. So these polymers which have the better sites, hydrophilic sites, are the polysaccharide derivatives such as cellulose, dextran, agarose, and polyacrylamide gel. These carriers can specifically have the hydrophilic groups that can specifically attach to the enzyme and can enhance the enzyme activity. So carrier binding can be afforded by three steps. First of all, we have, as we have discussed in the mucoadhesive, the adhesion properties. The adhesion of enzyme to a carrier sport can be dependent on the similar type of stickiness properties. If the carrier have a stickiness property, the enzyme have a stickiness property, they can attach to each other. This is known as physical adsorption. A physically enzyme can be adsorbed on the surface of the carrier. Secondly, we can develop a bond, an ionic bond, or we can share an electron, which is a covalent bond. If there is a spacer group, that spacer group will bind with the enzyme and the carrier by using ionic bonding or covalent bonding. Second method is known as cross-linking. We have discussed cross-linkers such as glutaraldehyde or the paraldehyde groups. Aldehyde groups can provide some more than one sites at which the second molecule can attach with each other. So if we use a cross-linker to intermolecular cross-linking of the enzymes or protein molecules or the functional group to an insoluble sport, it will be known as cross-linking. Cross-linking and enzyme cross-linking is a extensive reaction. Cross-linking can occur only when there will be a extensive reaction. A reaction or high reaction condition is required for cross-linking of the polymer. So the high temperature, high chemical reactivity of the cross-linker can destroy our enzyme's activity, and also this process will be more expensive and insufficient process that can result in the proteins malfunctioning. Generally, cross-linking can be used in conjunction with the other method. For example, we will load a enzyme into a gel and then we will cross-link that gel with the enzyme so that enzymes can be better stabilized within the gel. So a mean of stabilizing process is to preventing the leakage of the enzymes from polyacrylamide gel. For example, entrapped enzyme was in the polyacrylamide gel, and that entrapment can be stabilized by using cross-linking technique. So here is a diagrammatic representation of the cross-linking. The cross-linkers can attach one enzyme to another enzyme from one area to other area if we have protected the binding site. The bindings other than the binding site can be cross-linked to each other. So due to this severe activity, severe reaction condition, a cross-linker in which can proceed, it can result in the loss of activity. Secondly, if it is attached, cross-linker is attached very near to the binding site, then the molecule have the dimensions which are extensive than its binding site also. Extensive dimensions. That dimensions cannot fit to the surroundings and the enzyme activity will be lost. So a better technique from cross-linking is entrapping enzymes in a gel. For example, we have a matrix of a polymer. A matrix of the polymer means there is a gel-like matrix. For example, there are the attachment of the bonds of the polymer in which there is a specific porosity, specific pore size. And in that pore size, we can add the enzymes. And once the enzymes are added, they cannot be removed from that polymeric gel. So entrapment of enzyme is actually the localization of enzyme within the lattice of the polymeric matrix or polymeric membrane. It is done when the protein are attached to a substrate, and then the substrate and the gel polymer forming property is initiated. For example, we are using polysaccharides just as, for example, chitosan, and chitosan can be cross-linked or by ionic gelation method. So a protein can be entrapped within that ionically gelated chitosan molecules, and chitosan will result in the microparticles and the nanoparticles with enzymes. So enzymes are entrapped inside the gel, and that longitudinal crystals can be developed. That longitudinal crystals can be withdrawn from the solution phase also. So this type of entrapping enzymes are of two types. For example, a lattice type and microcapsule type, as it is evident from the pictures below. Matrix type means polymer and the enzymes are in 1:1 ratio. And in the membrane type, polymers are entrapped within the semi-permeable membrane. So lattice type means we have used some synthetic polymers such as polyacrylamide, we have used polyvinyl alcohol, we have used some natural polymers such as starch polymer or modified starch polymers. So they can develop into a cross-linked structure, cross-linked polymeric material when they are added to water or they are swollen. And they afford some interstitial spaces among the cross-linked water-insoluble polymer. So if we entrap our enzyme inside that lattice or interstitial spaces of the cross-linked water-soluble polymer, this type of entrapping enzyme will be known as lattice type. And if we can develop a semi-permeable membrane over the enzyme, then we can afford a microcapsule or nanoparticle of the enzymes. So microparticles can be grown to a layer structure that will result in the property of attachment, and they will not be soluble in the microparticle will not be soluble in the solution in which the reactant exists, so they can be centrifuged and recovered back from the solution if they are micropartically attached. So what are the advantages and disadvantages of this entrapping enzyme? The method जो entrapping enzyme है, it differs from the other cross-linking and covalent binding properties that enzyme does not bind in the entrapping enzyme process. But in the case of the covalent bonding, ionic bonding, adsorption, and cross-linking, there is a chemical reaction that will attach the enzyme to an enzyme or will attach enzyme to a sport. So conditions used in chemical polymerization are intense and severe. So any binding process which involves the reaction condition, it will be a severe reaction, and careful selection of the suitable conditions for immobilization are required. So there is a preferable method that we can use entrapping enzyme. So entrapping enzyme, one of the method most suitable method and most renowned method is microencapsulation or microcapsuling the enzymes. So for microencapsulation, we have discussed some methods in the topic of microencapsulation. One was interfacial polymerization method, in which the polymers can be developed in situ or polymers can be developed at the interface of the two immiscible liquids. So if the aqueous mixture of an enzyme and hydrophilic monomer are present in one solution and it is emulsified in a water immiscible organic solvent with one of the second monomer. So the monomers which are very hydrophilic monomer will attract the lipophilic monomer, and the monomers will adhere to each other at the interface. The interface of the aqueous solution in which enzyme is present with the organic phase in which a second monomer is present will be polymerized, and there will be a covering of the polymer over the enzyme. Polymerization will occur at the surface of the aqueous phase and at the boundary of the aqueous and organic phase. So this will result in the enzyme which is in aqueous phase is entrapped inside the polymeric membrane. Secondly, there is a technique in the entrapping enzyme is liquid drying. Liquid drying is also a microencapsulation technique. In this process, polymer is dissolved in water immiscible organic solvent. This is known as solvent evaporation technique, solvent dispersion and solvent evaporation technique. For example, we have dissolved a polymer in a water immiscible organic solvent which has a boiling point lower than the water and can be evaporated, and then the enzyme have been added to a aqueous phase or dispersed in the aqueous phase, and then these two organic solvent and aqueous solvent are emulsified, and then this emulsion can be heated so that the heat afforded by the emulsion can result in the removal of the polymeric solvent. Polymeric solvent, if the polymeric solvent is evaporated, the solid polymer will be deposited on the aqueous layer, and the aqueous layer will entrap the enzyme within that polymeric boundary that is developed at the interface. So this process can be afforded also by applying vacuum. If we applied a vacuum, the solvent can be removed at the lower temperature by heating at the lower temperatures. So we can protect the enzymes in microcapsules by freezing them and then developing a solvent remover method. There was another microencapsulation method known as phase separation or conservation. Phase separation means there are two colloidal solutions. For example, there is two hydrophilic colloids, or there are one organic and one aqueous colloid. When they are mixed with each other, they have the affinity with each other. So that affinity will result in the separation of the dissolved materials or polymeric materials from the solutions. So if a polymer which is dissolved in the solution will be separated from that solution, it will fall on the enzyme surface, and enzyme surface will and on enzyme surface they will be cross-linked to each other to form a micro particle or a layer around the enzyme system. So this type of method is known as conservation, and conservation can be simple, conservation can be complex, and conservation can be organic also. You can afford an organic conservation, you can salt out the polymeric material from the organic solution, or you can salt out from the aqueous solution. And if only one hydrocolloid is being used, then it is simple aqueous conservation. And if two hydrocolloids are used, then it will be complex aqueous conservation. What are the applications of the immobilized enzyme? There are the two main bioreactors in the bioremedication. There are the suspended growth and the fixed film. So enzymes can be used in bioremediation. So bioreactors with suspended growth and fixed film can be used. Suspended growth bioreactors consist of batch, plug flow, and completely mixed reactors. Microorganism can be suspended in the medium of the reactor, and the enzyme can be passed through that reactor. So this will be suspended growth bioreactor. Fixed film bioreactor consists of fixed bed. It will have a fixed bed or a fluidized bed or air sprayed bed. Here microorganisms grow on a or within the solid medium in the reactor. So in the reactor in which we have attached a enzyme, the microorganism will grow within that solid medium or reactor. So it is a fixed type of. A second method is application of the enzyme is detection of heroin and its metabolites. Normally athletes which abuse some drugs and they can increase their performance during the sports activity, we can detect these metabolites which are metabolites which can enhance their sports activity by using some enzymes. If we withdraw the blood of the athlete and then treat it with the enzyme which convert those enzymes into the specific products, or enzymes can react to those products. Enzymes are very specific which can only react to the heroin and its metabolites, for example morphine and its derivatives. So if there will be morphine or its derivatives, they will react with that enzyme and they will be converted into product. And enzyme reaction, we can check that enzyme reaction. So according to the National Institute of Drug Abuse USA, it accepts 0.1 milligram per ml morphine as positive result in urine analysis. So if 0.1 milligram per ml reaction occurs with the enzyme, it will be called as positive reaction. So it will be called that athlete or the person have used in the recent past a morphine or the product of the morphine. It is believed that it could be lower to estimate can be lower to 0.05 milligram per ml, or our detection limit is mostly 0.1 milligram per ml. NIDA and other agencies around the world desire a more sensitive test than this blood test to detect the morphine and heroin in the blood samples. This approach, which is used by the NIDA, is the enzyme known as acetylmorphine carboxylesterase AMCE and other morphine dehydrogenase MDH enzyme. They have been isolated from the bacteria and they have been immobilized, and then they are reacted with the blood solution blood products of the or the serum products of the humans or the athletes, and if there is a reaction, there is any indication of the reaction that heroin or morphine or derivative of morphine have been reacted with that enzyme, then that will be reported as positive reaction. So last example is isomerization of glucose to fructose. Glucose isomers, D-glucose, ketoisomerase. This is the enzyme D-glucose ketoisomerase. It causes the isomerization of glucose to fructose. So in a patient which is a diabetic patient, a sugar patient, we can convert all the glucose contents into fructose, and that fructose will be converted into glycogen. And if the fructose, which is twice sweeter than sucrose, can be used as a sweetening agent, the process will be more facilitated for a sucrose to be used as a sweetening agent or a fructose to be used as a sweetening agent. So half of the sucrose will be required to get sweetening effect if we are using fructose. So the process of conversion of D-glucose into fructose by using D-glucose ketoisomerase enzyme will help isomerization of the glucose to fructose.